Welcome students to today's lecture on molecular biology. Today, we will be delving into the fascinating world of ribosomes and their role in various cellular processes. Ribosomes are composed of ribosomal RNA, RNA, and proteins, and their main function is to synthesize proteins through a process called translation. Now, let's move on to our first topic, ribosome biogenesis. Ribosome biogenesis involves the synthesis, assembly, and maturation of ribosomal subunits. It begins with transcription of RNA genes, followed by processing and modification steps. Ribosomal proteins associate with RNA to form subunits, which then join in the cytoplasm to form functional ribosomes for protein synthesis. Cryo-electron microscopy, cryo-EM, is a technique that freezes samples to preserve their structures. By directing a beam of electrons at the frozen sample, two-dimensional projection images are produced. These images are combined to generate a three-dimensional density map of the ribosome. Advances in cryo-EM technology have revolutionized ribosome structure determination, revealing intricate details about their structure and function. Cryo-EM has also provided insights into dynamic aspects of ribosome function, such as the movement of tRNAs and ribosomal translocation during translation. Ribosomal RNA modifications will be discussed next in relation to translation fidelity, in the section on 3. Ribosomal RNA modifications and their impact on translation fidelity. We will discuss the covalent alterations made to RNA molecules in ribosomes. These modifications, such as methylation and pseudouracilation, are crucial for ribosome assembly, stability, and accurate translation. Ribosome-associated factors regulate ribosome function. Ribosome recycling factors like RRF and FG recycle ribosomes. Quality control factors like RQC and ZNF598 detect errors during translation and degrade or release stalled ribosomes. These factors maintain translation regulation and quality control. Ribosome hibernation is a process by which ribosomes enter a dormant state to cope with stressful conditions, such as nutrient deprivation or environmental changes. During hibernation, ribosomes undergo specific modifications to allow them to remain inactive until favorable conditions are restored. One of the key modifications involved in ribosome hibernation is the binding of specific hibernation factors, such as HPF, hibernation promoting factor, onto the ribosome. These factors induce conformational changes in ribosomal proteins, shielding the ribosomal subunits and preventing them from engaging in translation. Ribosome hibernation is a reversible process, and upon the restoration of favorable conditions, the hibernation factors dissociate from the ribosome, allowing it to resume translation. The study of ribosome hibernation provides insights into how cells adapt to stress and ensure their survival under adverse conditions. It also highlights the versatility and adaptability of ribosomes in response to changing cellular environments. Now, let's move on to our next topic, non-canonical ribosomal functions. In this section, we will discuss how ribosomes can pause or become stuck during protein synthesis and the mechanisms that cells employ to rescue and resolve these stalling events. Ribosomes occasionally encounter obstacles during translation, leading to translational errors and protein misfolding. Cells have evolved rescue mechanisms, like tRNA and ARFA, to rescue stalled ribosomes. These mechanisms ensure translation fidelity and prevent the accumulation of defective proteins. In this section, we will explore the various post-translational modifications that ribosomal proteins can undergo, how these modifications affect ribosome structure and function, and the potential implications for cellular processes. In summary, ribosomal proteins undergo various post-translational modifications that regulate ribosome function. Phosphorylation, ubiquitination, acetylation, methylation, and sumoylation are key modifications that impact ribosome assembly, translation, and protein interactions. These modifications provide insights into ribosome regulation and cellular signaling. Next, we will discuss ribosome profiling and its applications in understanding translation dynamics. Ribosome profiling, or RiboSeq, is a technique that captures ribosome positions during translation. It provides a genome-wide view of translation dynamics and helps understand how translation is regulated. It has uncovered new regulatory mechanisms and can be integrated with other omics data for a comprehensive understanding of gene expression. Ribosome-targeted antibiotics selectively inhibit bacterial ribosomes, halting translation and inhibiting bacterial growth. Different antibiotics act at different ribosome sites, blocking elongation, macrolides, distorting decoding, aminoglycosides, or inhibiting peptidyltransferase activity, tetracyclines, chloramphenicol. Understanding these mechanisms aids in fighting antibiotic resistance. Evolutionary conservation and diversity of ribosomes across species is our next topic. Ribosomes are essential molecular machines found in all living organisms. They are highly conserved but also exhibit species-specific variations. These variations affect ribosome-mediated processes and can impact translation efficiency and accuracy. Ribosomes can also adapt to different cell types and conditions. Comparative analysis of ribosomes has helped understand their evolution, disease-related defects, and potential therapeutic interventions. In conclusion, our study of ribosomes has revealed their multifaceted role in cellular processes, from protein synthesis to intricate regulation and adaptation. This knowledge offers promising possibilities for medical interventions and combating antimicrobial resistance. Thank you for joining me today.